Dear colleagues, dear friends, it is about how molecular modeling can predict a gene function, reveal evolutionary constraints and opportunities of the gene, and what molecular modeling think about the true amino acid substitution matrix. All of this will be about legume-resolvable nitrogen fixing symbiosis because it is a perfect evolutionary model demonstrating coevolution between two tightly integrated gene systems. And the first step of interaction between rhizobia and legumes is a signal exchange. At this stage, partners recognize each other and launch rot nodular formation and nitrogen fixation. From rhizobia side, the system consists of genes controlling node factor synthesis. It is a signal molecules. From legumes side, it is a system of plant receptors responsible for node factor perception. And what molecular modeling can, how molecular modeling can predict a gene or prove gene function. And plant receptors for node factor perception belong to a family LISM domain containing receptor-like kinases and contain 2 or 3 LISM domains in the extracellular part of the protein, transmembrane domain, and intracellular kinase domain. All plant receptor kinases fall into two classes: leak with active kinase and clear with not active. Usually, plant receptor acts as a heterodimer consisting of two receptors, one leak and another lear. And there is mystery of the Afghan peas. It's a very long story, and I'll try to tell about it very short. There are two lines of peas, European Linus and Afghan Linus, and both of them belong to the same pea species, Pisum sativum, and rhizobia isolated from each Linus belong also to the same species, Rhizobium leguminosarum. But there is a difference in symbiotic performance of rhizobia isolated from different hosts. Rhizobia isolated from Afghan peas can produce nodules on both Linus, on Afghan peas and European peas. But rhizobia isolated from European peas can produce nodules only on European peas. And this secret is not secret now, because the problem is acetylation, not factor. Rhizobia isolated from Afghan peas have not X gene responsible for acetylation, not factor. Because of this acetylation, we see this differential reaction with different lines. But if from the rhizobial side is quite clear what the problem, but the problem was in plant side, because people know that there is some receptor gene responsible for perception of, responsible for differential reaction with acetylated and non-acetylated signal. But until now, the gene called SIM2 have not been cloned and sequenced. I will skip all this matter, and at the point we started work with this hypothesis, there were three candidates on to be three candidates to be SIM2 receptor: SIM37, K1, and LEAK-X, and we decided to check it with molecular modeling. And due to specific, very specific polymorphism pattern, the LEAK-X gene was the most suitable candidate for SIM2. And actually, we used power of molecular modeling to check if the heterodimer containing LEAK-X gene demonstrates differential reaction with acetylated and non-acetylated node factor. And finally, we demonstrated that LeakX is a perfect candidate for SIM2, explaining the differential interaction with acetylated, non-acetylated factors. Exactly the same picture as we see on this picture demonstrating nodal formation with different type of rhizobia. So it is very interesting, but in this case, molecular modeling can predict or prove and help to find which of the genes is responsible for perception of, for different perception of acetylated, non-acetylated node factors. And all the details of this study are available in this article. Another thing I'd like to discuss now is how molecular modeling reveals evolutionary constraints and opportunities of the gene. It is a very small field in Gachina district, not far from St. Petersburg, but there is extremely high diversity of wild growing legumes. And actually we did a lot of very interesting works on this field. And 2 years ago on Biata 2090, I reported one of them. It was about evolutionary molding concept, the introduction of a new type of diversity, topological beta diversity, an effect of gender oscillation, and red queen, red king model. All of these things are available except gender oscillation in this paper. But now I'd like to show you another work originated from this field. We collected on this field a very big DNA pool from Vicia plants. It's also legume plants capable of nitrogen fixation, and it has the same receptor system. And we tried to study amino acid polymorphism in heterodimer receptor system. And we prepared 2 random cloning libraries with this lithium domain containing part of the 2 genes SIM10 and K1. It was about 100 sequences in each libraries, random sequences. And we built molecular model of the receptor dimer, and we formed contact zone between 2 receptors. Then we mapped all polymorphic sites identified from analysis of clonal libraries, and made projections of this polymorphism on the contact zone, and perform node factor docking. And you can see on this picture node factor docked and contact zone between 2 receptors with polymorphic sites mapped. It's a bilateral polymorphism, and with red you can see polymorphic site originated from NFR5 SIM10, and with green from K1 part of this receptor. It is very interesting because we can see how polymorphic zones distributed in context of node factor chemical structure. And we see that the zones bilateral polymorphism marked with yellow is near the very important part of node factors responsible for variation in host specificity. And this picture suggests that there is intra-population diversity of chemical structure, not factors, and probably indicates presence a co-adapted pair of micro and macro symbionts. Also, we tried to map selection pressure and contact zone in different part of these molecules, and I think it is very interesting thing. But all the details of this study are available in this paper. And finally, I'd like to discuss what molecular modeling thinks about the true amino acid substitution matrix. You know that traditional amino acid substitution matrices, PAM, GTT, BLOSUM, are kinds of averaging of evolutionary effects. But in this context, when we work with molecular models, we can see that the same time of substitution, for example, serine to tyrosine, in different contexts, may have absolutely different effects on the end function. For example, effectivity interaction with node factor. And with molecular modeling, we have a perfect tool which allows us to calculate the effect of any substitution in any context and build the true substitution matrix. Just imagine what it can be. We think that the true substitution matrix should be first, specific for each particular protein, the second, specific, of course, for each ligand or substrate, and three, of a complex structure, for example, constructed as a stack of partly filled square matrices, one matrices per each amino acid position. Partly filled because, of course, there is prohibited states, but it is the image of or fantasy of how the true substitution matrix can be built. But do we really capable of building this matrix or not? I think not, because another very important problem is it is epistasis, which demands for building a separate matrix for all initial variants of the protein with substitution in other position. And I think it is impossible task. And to resolve it, probably we have to call for Demon's help. I mean Demon's in good sense. It's demons from this company, you know, Maxwell demon, Descartes demon, Laplace demon, and some other demons not so known, like Nietzsche demon, Darwin demon, and probably there should be a demon of the matrix who could build this true substitution matrix. And with this correction, that the last topic of my presentation should sound like that. Molecular modeling thinks about the true amino acid substitution matrix and calls a demon for help. Thank you very much for attention, and many thanks to my colleagues Anna Igolkin, Yaroslav Solovyov, Yuri Porozov, Zhukov Vladimir, Sulim Anton, and Nikolai Provorov. Thank you very much.